Start. Yeah, so welcome everyone on this lovely sunny St. Patrick's Day. We're excited to have you here, and we're very excited to have Dr. Christopher States presenting for us today. Before I introduce him, I'd like to first make you aware of some upcoming events that we have planned, and Kara's going to provide links to them in the chat box. Of course, we have our wonderful environmental health series that's continuing on. There's an Earth Day, John P. Wyatt Symposium coming up, which will be on Earth Day, April 22nd, which participation will be from all the great environmental groups and centers across campus pitching in for this big event, and the keynote is Howard Frumkin. So we look forward to you participating in that. So the announcement should be sent out, but please mark your calendars. Earth Day, April 22nd. It starts at noon and it'll run till 3. All right. So thank you everyone for joining. It is my absolute pleasure to introduce our guest today, Dr. Christopher States. He is joining us from the University of Louisville, just down the street. We were just discussing how our centers he is the director of an environmental health center there in Louisville. And we're discussing how our centers could collaborate for the betterment of the Commonwealth. And I'm just so excited that Louisville has a center. Dr. States is the vice chair for research in the Department of Pharmacology and Toxicology. He's also the associate dean for research. And as I mentioned, he is the director of their environmental health center, which is called Center for Integrative Environmental Health Sciences. And today he's going to talk about his research focus, which is microRNA dysregulation and aneuploidy in arsenic-induced carcinogenesis. So Dr. States, welcome. We're delighted to have you, and thank you for joining us today.
Okay, well, it's my pleasure. Thank you for the very kind introduction and the invitation. And I see a familiar name or two in the participant list. Good to see you all again. Been a while since I've been out in Lexington. So today, I'm going to talk to you about some work we've been doing the last couple of years. And let's see if I can get the slide to advance. So I have no commercial conflicts of interest, and the work's been supported by NIEHS grants, and we had a visiting scholar from China in for a while, and he was supported by Homeland funding from there. And our biostatistician has an endowed chair. So let's try this again. All right, so first I'll give you a little background, talk about environmental arsenic exposure, talk about chronic arsenic exposure and cancer, and I'll get you all oriented on microRNAs and their function, so we all have a basic understanding. To go then into studies we've done on microRNA dysregulation, and then the impact of a particular microRNA on chromosome stability, and then talk about the potential mechanism for the overexpression of this microRNA, and a little bit on some future directions and conclusions. So key points, take-home points. Arsenite transformation of keratinocytes induces chromosomal instability. And chromosomal instability is a paradigm of cancer. Just about every cancer that's ever been looked at exhibits chromosomal instability. HSA miR186, which will be a main topic, I'll show you that it's elevated in arsenic-induced squamous cell carcinoma, and that this microRNA induces chromosomal instability that is further enhanced by arsenic co-exposure in an in vitro model. And then I'll talk about that it may be induced as a consequence of arsenite disruption of ZRAMB2 function, ZRAMB2 being an apical splice factor. So arsenic is known as the king of poisons and the poison of kings. So very long history of use for homicide, which is probably what comes to everybody's mind when they hear arsenic. And that's because it's tasteless and odorless. So famous examples of a victim is Napoleon Bonaparte is thought to have been a victim, and Cesare Borgia is thought to have been a major perpetrator. So he was a medieval Renaissance Italian royal who got rid of his rival's by poisoning. But currently, and what we're most concerned about is rather than the acute poisoning, we're talking about chronic low-dose poisoning, and that's mainly by chronic ingestion of arsenic-contaminated drinking water, and the largest mass poisoning in history now, with more than 30 million alone in Bangladesh. So what is arsenic? So it's an element, and it's a metalloid right here. So this line here are those elements that are non-metallic by valence, but they oftentimes behave as metals. And oftentimes arsenic is found in nature as an oxyanion, so arsenite or arsenate. It's very widely distributed, and it causes multiple diseases. It's the 20th most common element in the Earth's crust. I have more than 200 million people worldwide that are exposed to arsenic in their drinking water as a result of natural contamination. It's been #1 on the ATSDR National Priority List of hazardous substances for more than 20 years. So these are substances of concern at Superfund sites. And what I find most interesting about it from a toxicological point of view is that there's a variable disease outcome. So it's a known human carcinogen. It's a known human athergen, that is, it causes cardiovascular diseases, and it's a probable human teratogen. And now the newest evidence implicates it in diabetes as well. So here's the heat map of arsenic in groundwater worldwide, where the red areas are the highest. And the white areas generally are not no arsenic, but insufficient data. And those same parameters apply to this heat map of arsenic in groundwater in the US from the US Geological Service. This is a map by county. So the red areas are where the arsenic in groundwater is greater than 50 ppb, which was the old standard prior to 2001. The current standard is 10 ppb, and the orange areas are above 10 ppb. Once again, the white areas are not no arsenic, it's insufficient data. And what's the importance of this is that private well water is unregulated by the EPA. And EPA only limits arsenic in public water supplies, and only in public water supplies that serve better than 4,000 people. As a consequence, there's over 2 million people in the US that are exposed to arsenic in private well water. And around the time I became interested in it, I was living in Detroit, working at Wayne State University, and this article came out in the Detroit Free Press about this unfortunate gentleman who was the first confirmed case of arsenicosis from well water in the US. And he lived in the thumb of Michigan here. And this dark area is the area that geologically is predisposed to arsenic containing minerals that then leach out into the water. So I do like to bring things home to folks. And so is it in Kentucky? Yes, it's in Kentucky. And you'll notice, once again, the red areas are 50 ppb and above, and the orange areas are 10 ppb and above. And you notice that the high concentrations are where there's a lot of coal, right? So arsenic is associated with coal, and consequently, arsenic is released into the atmosphere when coal is burned. And so here on the left is a picture of a woman in rural China where she's using coal indoors, well, kind of, for her cooking. And so she's exposed to coal smoke that contains arsenic, and arsenic is endemic in this area as well. And here's a picture of a power plant here in Kentucky and burning coal, and water pollution's often around that. And they store the coal ash either in slurry ponds or ash piles adjacent to the coal-fired power plants. And those tend to leach out into the groundwater around the area. And so here's some photos taken from this article from Ryan Van Velser of WFPL, that's the public radio station here in Louisville, of leaching from the ponds and coal ash piles from coal-fired power plants. So it's a serious environmental problem. It does get into the water, and we have some information from some dermatologists that they've actually seen arsenical skin lesions, which we'll talk about shortly here in Kentucky. So it's not only in drinking water, it's also in food. The two main culprits are rice and fruit juices. And this is because arsenicals were used as pesticides for cotton, right? And also in orchards. And so what's the connection between the use on cotton and rice? It's that cotton production in the United States in particular is way down, but they're now using those fields for rice production. So rice produced in the south central US, Texas, Louisiana, Arkansas, has higher levels of arsenic in it than rice from other areas of the world. And the rice is one of those plants that bioaccumulates arsenic. The other is tobacco. So the other place that arsenicals were used was on tobacco plants throughout Kentucky and other areas where they grow tobacco, and those fields are heavily contaminated with arsenicals. Arsenics also was in treated lumber that was used residentially. It's been banned from residential use, but it's still used for commercial use, non-residential use. And a lot of the playgrounds that are around that have treated lumber in them, they're treated with arsenicals as well. So that's CCA treated lumber, which is copper, chromate, and arsenate. So three baddies. So chronic arsenicism is the disease state that results from chronic ingestion of low levels of arsenic. And some of the symptoms are peripheral neuropathy, hyperkeratosis. You can see in this picture borrowed from the New York Times, these nodules on the palms. They also occur on the trunk, but predominantly on the palms and soles. Those are hyperkeratotic lesions, and those are premalignant lesions. Bowen's lesions, which are squamous cell carcinoma in situ. Diffuse melanosis, so there's a lot of darkening of the skin. Arterial sclerosis, which results in a dry gangrene predominantly of the digits. And you can see this man has lost a finger, and that's due to the dry gangrene. And it also can cause a wasting syndrome. So the arterial sclerosis was first observed in Taiwan, and it was named Blackfoot disease. And you can see why. This is a picture from an individual suffering from the disease in Taiwan. And this, once again, is due to the dry gangrene due to arterial sclerotic disease, not unlike what is seen in diabetics. So it's thought to be specific to Taiwan for quite a while. And then when the problem erupted in Bangladesh, the same problems were seen in Bangladesh. So we know that this arterial sclerosis is not due to something specific to Taiwan, but is a widespread problem caused by arsenic ingestion. And arsenic also causes problems, including myocardial infarction, stroke, kidney disease, other diseases that are result from arterial and atherosclerosis. But this talk is going to be about cancer, in particular squamous cell carcinoma. But in addition to squamous cell carcinoma, chronic arsenic ingestion causes basal cell carcinoma, another form of skin cancer, and bladder cancer and lung cancer. Those are the leading cancers caused by arsenic. But there's also strong evidence that liver, prostate, kidney, and ovary also suffer carcinogenesis from chronic arsenic ingestion. But we're going to focus on squamous cell carcinoma for the time being. So there's multiple mechanisms that have been proposed over the years for arsenic-induced carcinogenesis. These include DNA repair inhibition or co-mutagenicity, epigenetic modulation, DNA methylation, differential microRNA expression, oxidative stress. Anytime you see metals, oxidative stress is going to be invoked. Changes in cell proliferation and stem cell expansion. It's another mechanism that's been proposed. Differential alternative splice or mRNA splicing, there's evidence for that. And aneuploidy or increased chromosomal instability, which is aneuploidy being numerical and structural chromosomal abnormalities. So more than 20 years ago now, the National Research Council convened a committee to review the literature and come up with mode of action recommendations. And at that time, and this is a direct quote, the most accepted explanation for the mode of action for arsenic carcinogenicity is that it induces chromosomal abnormalities without interacting directly with DNA. So it's genotoxic, but it doesn't cause point mutations like most of the chemical carcinogens that we're used to studying. So the NRC made this recommendation, and virtually no one investigated chromosomal abnormalities with arsenic exposure. They have followed all those other mechanisms that I listed on the previous slide. And indeed, there's evidence for all of these things. But what I'm going to try to show you is that dysregulation of microRNA expression is intimately linked with the induction of aneuploidy or chromosomal instability, inducing both numerical and structural chromosomal abnormalities. So in the 90s, there were a number of studies that showed that arsenic and arsenite in particular disrupts mitosis and induces aneuploidy, and this is a variety of cell systems, including diploid cells. And a model was developed for in vitro model for studying arsenic skin cancer, arsenic-induced skin cancer, which is because exposing laboratory animals to arsenic in their drinking water does not induce cancer in an adult animal. The only way to induce cancer in a mouse with arsenic exposure is prenatal exposure. And in that case, they do not still develop skin cancer. So there's no in vivo model for arsenic-induced skin cancer other than the unfortunate humans that have been exposed. So in vitro model is chronic exposure of an immortal cell line haycat to low levels of arsenite, that is 100 nanomolar sodium arsenite, which corresponds to the blood level observed in people exposed to 100 to 300 ppb arsenic in their drinking water, which is a fairly common exposure in the disease-prone areas. And so this experiment was done with 4 cultures each with 0 and 100 nanomolar sodium arsenite. And after 28 weeks, you can see that the exposed cells had a higher fraction of cells with increased chromosome number. All right, and so that's numerical aneuploidy. So I'm going to diverge momentarily and talk a little bit about microRNA structure and function. So microRNAs are short, non-coding RNAs that regulate gene expression post-transcriptionally. And they do that by interacting with messenger RNAs. They are processed from large RNAs that are transcribed by RNA polymerase II, either as microRNA-only genes or embedded in introns and protein coding genes. So RNA polymerase II is the polymerase that we learned about in biochemistry that transcribes protein coding genes. We now know that they also transcribe these microRNA-only genes, and microRNAs also can arise from introns and protein coding genes. So they are excised from these long primary transcripts in several stages. There's the prime microRNA initially, which is trimmed down and then exported, and that's processed further where either the plus or minus the 5' or 3' strand then binds with argonaute to form the RISC complex bound to the mRNA where it does its function of inhibiting translation. So many microRNAs have been shown to play a role in multiple cancers. This is actually an old slide. If I put new data on here, it would completely fill this slide. The point is that there's a lot of work showing that dysregulation of microRNAs plays a role in carcinogenesis. And so how do they work? So you may have inferred already, they work opposite to their expression level. So association between a microRNA expression and tumor development and progression and response to therapy are used as biomarkers for disease diagnosis, prognosis, and therapy. And they work opposite, as I mentioned. So an oncogenic microRNA, when it's overexpressed, will suppress a tumor suppressor gene. Likewise, a tumor suppressor microRNA, when it's underexpressed, will relieve suppression of an oncogene. So understanding microRNA function and trying to put together the pieces between the expression of microRNAs and their target mRNAs is somewhat complex. So another version of the exposure map, just highlighting the major areas. And we're going to focus in on the Ganges Delta, which is where Bangladesh is. And right adjacent to Bangladesh is the western state of West Bengal of India. And so our collaborator there, Dr. Ashok Giri, who's at the Indian Institute of Chemical Biology, was kind enough to provide us with some samples of arsenic-induced skin cancers and premalignant lesions from this area right here in red, which is the western part of West Bengal, right up against Bangladesh. And this is the arsenic endemic area in West Bengal. So these people are exposed to high levels of arsenic in their well water, and they exhibit these skin lesions. Here's the hyperkeratotic lesions. There's a gross and histological figure. And what's interesting about arsenic-induced skin cancer is that there is a known premalignant lesion, the hyperkeratosis, which can give rise to either basal cell carcinoma or squamous cell carcinoma, passing through what's known as Bowen's disease. And as I mentioned earlier, Bowen's disease is squamous cell carcinoma in situ. And what's unique about arsenic-induced cancer is that people will have multiple hyperkeratoses, multiple Bowen's lesions, and they can also have basal cell carcinomas all at the same time. Whereas usually with UV or sunlight-induced cancer, you see one type of cancer and only one tumor at a time. So we're going to focus here on hyperkeratosis to squamous cell carcinoma pathway. And what we did is we used laser capture microdissection to dissect the keratinocyte cells from the thin sections of these lesions, but hyperkeratosis and either Bowen's disease or squamous cell carcinoma, and purified the RNA from those laser capture microdissected cells, and then used the cancer panel from Invitrogen for RT-qPCR arrays to look at microRNAs that were expressed. And this work was actually done by my daughter when she was working for Dr. Susan Galandiak on a similar project in colon carcinoma. And so in the first two squamous cell carcinomas that she analyzed versus the two hyperkeratoses, these are the mean fold change. And HSA miR186 was at the top of the list by far as being overexpressed in the malignant squamous cell carcinoma as opposed to the premalignant hyperkeratosis. And so at that time, what was known about HSA miR186 is that it targeted securin, which is also known as PTTG1. And this was validated in this paper here. And here's the predicted binding. So securin's an interesting protein. Securin is an essential protein, and its job is to regulate separase activity. Separase is a protease that cleaves the cohesins that hold the chromatids together in a chromosome. So to separate the chromatids, you have to activate separase, you have to degrade securin. And so in earlier studies, we had seen that arsenic exposure halted cells in metaphase, and that there was an accumulation of cyclin B, and securin. So both of these are degraded by the in the ubiquitin pathway by the E3 ubiquitin ligase, the anaphase promoting complex, and then directed to the proteasome for degradation. And we saw them accumulated. So it appeared that the arsenite was inhibiting the APCC. And we have a project going on now looking at the E3 ligase subunit involved in that, which is a zinc finger protein. So we also had worked out that the arsenic exposure and the prolonged mitotic arrest that occurred induced P53, and P53 in turn induced CDKN1A, which is also known as P21. And we showed that that induction of P21 was essential for the cells to progress from metaphase to anaphase, escaping that block. But what we were not able to figure out at the time was how securin accumulation could be prevented. And so seeing overexpression of miR186 suggested a mechanism, which is that if miR186 is overexpressed, the securin levels will be lowered, And so you need less APCC activity to get rid of what's there. So securin, I like to think of it as a Goldilocks protein. You need just the right amount of securin to be present. So too much securin, as in inhibition of the anaphase promoting complex by arsenic exposure, can inhibit chromatid separation, which will lead to lagging chromosomes and aneuploidy. Too little securin, on the other hand, as in excess miR186, can lead to premature chromatid separation, leading to chromosomal instability and aneuploidy. So around the time that we had observed the increase in miR186 expression in arsenic-induced squamous cell carcinomas, I was contacted by Dr. Jigwa Wu, who's an associate professor of public health in Guangzhou. And he wanted to come to my lab and do something with microRNAs and arsenic. And I said, have I got a project for you? So the project that he worked on was to overexpress miR186 in the HACAT model and see what happened. And so what he did is, at that time, we had all that was available was an overexpression plasmid. So he took that plasmid and he transfected that into HACAT cells in isolated colonies, or we deleted the microRNA coding cassette to make an empty vector control and made colonies with that as well. And so then he characterized those colonies for miR186 expression, and these are the miR186 expression levels in the colonies he analyzed. And then he picked the 3 top expressing clones from the miR186 transfected and the bottom expressing from the empty vector for further study. And what he did is he continued, then he split those clones into 2 cultures each, one which was exposed to 0 and another that was exposed to 100 nanomolar sodium arsenite. And he incubated those for up to 8 weeks. And these are the data looking at miR186 expression in those clones over that time period. On the left are the 4-week samples, and on the right are the 8-week samples. And what you can see is that in all cases, the overexpression was maintained in those cells, those clones transfected with the miR186 overexpression vector, and it remained low in the vector control transfectants. And in all cases, the cells transfected with the overexpression vector had higher miR186 expression than the control vector. So he then analyzed those clones at 4 and 8 weeks to look for chromosomal abnormalities. And initially what he did was simply count chromosomes in metaphases, and also looked for dicentrics. I don't know if you can see it on your screens, but these are chromosomes with 2 centromeres. And he also counted ring chromosomes. So these are chromosomes in which the telomeres have been lost and the ends have fused, so they're now a ring. And then double minutes are teeny tiny dots, which I'm sure you can't see. And double minutes are chromosome fragments that were associated with gene amplification as long ago as when they were studying amplification of the dihydrofolate reductase gene in methotrexate transfected treated cells. And now it's known that they're associated with oncogene amplification. So what you can see in this slide, so HACAT cells are aneuploid, they're immortal, and almost every immortal cell line that are available, you'll find are aneuploid. So they start out with about 77 chromosomes. What Jigwa saw was that a fraction of those chromosomes in the miR186 expressing would appear with lots of chromosomes, 150, 250 chromosomes. And also he saw the induction of these dicentrics, double minutes, and rings. So he quantified those with the help of Dr. Sandy Wise, who joined us around that same time. Dr. Wise is a certified cytogeneticist, so she brought that expertise to pharmacology. We're going the wrong way. Okay, and so first, what we called things that had more than a 10% increase in chromosomes supernumerary. And so you can see that overexpression of miR186 induced supernumerary chromosomes and that at later times this was enhanced by concurrent arsenite exposure. And in particular, I already mentioned that there were mitoses that had more than 240 chromosomes. And the only way that to arrive at this is to have multiple endomitoses. So first, doubling the DNA to about 150 chromosomes, and then doubling them again to 300 and shedding of some chromosomes. So we had to have cells that had undergone an endomitosis survive and then go on to become even further aneuploid. And that's truly remarkable in human cells. So dicentrics also were increased in miR186 overexpressing cells and were significant at 8 weeks, and some possible modest increase in them with arsenite exposure. What was most interesting was the double minutes. Double minutes are definitely increased with miR186 overexpression and further increased with arsenite exposure. So this is evidence, strong evidence of structural aneuploidy as well. And ring chromosomes were only seen in cells overexpressing miR186, but because of the low numbers, there's no statistical significance. So since then, so unfortunately, Dr. Wu had to return to China as his mother became ill with ovarian cancer and was unable to continue the project. But last year, a graduate student joined the lab, Agaliki Likudi, and she has a strong interest in cytogenetics. And so she's been working with Sandy Wise as well now to do karyotyping on these cells. And these are some examples of abnormalities that she has observed. So marked here in red are marker chromosomes that are known translocations in HACAT cells that help to identify the cells as HACAT. And then in red, the red arrows mark instances of, the blue arrows are instances of chromosomal instability induced, and that's either a translocation or a duplication of a chromosome. So she's gone on to some preliminary data. So she's only analyzed 15 metaphases each so far of each of these clones. But what I wanted to show you these because they're showing clear differences in the patterns. So these are abnormalities in chromosome 1, 11, or 20, or the marker chromosomes, and double minute chromosomes. And what you can see here on the left are the vector control cells. On the right are the miR186 overexpressing cells. On the top are no arsenic, and on the bottom are the 100 nanomolar exposed cells. So you can see an immediate difference in the pattern between vector control and miR186 overexpressing. What you can also see is that when arsenite exposure is included, you start to see an increase in double minutes. So clearly, there's an interaction going on both from miR186 and arsenite exposure. And further analysis explains why the statistical problems we've encountered. So each of these clones appears to be behaving in a completely independent fashion. So if you think about what's present in a tumor, it's well known that tumors are not homogeneous, that they're groups of independent clones of independently evolving cancer cells. And we're actually seeing this in this experiment in the 3 clones that have been analyzed in each group. And you can see here, the vector controls are all pretty tight, whereas the ones that are overexpressing miR186, we're seeing a lot of variability in the chromosomal abnormalities that are seen both in the unexposed and arsenite exposed cells. So we're seeing stochastic behavior in this model system reflective of what actually goes on in a tumor. So to summarize this part of the talk, I've shown you that arsenite exposure enhances chromosomal instability, that HSA miR186 is a microRNA that's overexpressed in squamous cell carcinoma relative to premalignant hyperkeratosis and arsenic-induced disease, and that overexpressing miR186 in the HACAT model induces chromosomal instability with both increased chromosomal numbers and increased structural abnormalities, i.e. aneuploidy in both cases, and that these are enhanced by arsenite exposure. So we were interested in what pathways might be targeted by HSA miR-186 and what might be going on besides an impact on securin. And so we used a program called MIRPath version 3, which is available on the web publicly. And what this program does is it takes the interactions that are cataloged in tarbase, or one of two other microRNA databases, and takes the targeted mRNAs and overlays them on the KEGG pathways. And this is the results of the tarbase targets of HSA miR-186-5P, which we know from some RNA-seq data is the major, it's almost exclusive miR-186 form that's expressed in HACAT cells. And number 6 on the list is cell cycle. And so we're very interested in seeing what that had to say. And so don't worry about trying to read what's in the little boxes. What's important to see here is the yellow boxes are the predicted targets of miR-186. And this is a cell cycle pathway starting over here in G1 control, and over here is mitotic control. And so this group is the spindle assembly checkpoint, which is what activates a mitotic arrest in response to dysregularities. And here is the anaphase promoting complex, two members of the anaphase promoting complex, cyclosome. And so what we saw was that BUB1 and CDC27 were predicted targets. So we're interested to see if anything was actually happening to those proteins in the clones that Jaguar prepared. And so this was picked up by a postdoc in the lab, Anna Cardoso. And these are some Western blots that she did on 8-week samples from Jaguar's clones and looking at BUB1 and CDC27. And we'll walk through that. First, the BUB1. And what you see is that miR-186 overexpression reduced BUB1 levels significantly in the arsenic unexposed cells, but arsenic exposure seemed to induce that expression and seemed to mitigate the reduction induced by miR-186. So something very complex is going on with BUB1. BUB1 is a component of the mitotic spindle assembly checkpoint. CDC27, on the other hand, was unchanged. So BUB1 clearly is targeted by HSA miR-186, and it's induced by sodium arsenite. CDC27 was not suppressed by miR-186 or overexpressed or changed by sodium arsenite. So that raises a number of questions for this protein. Is the prediction incorrect? So once again, these are predictions. Are there other factors modulating the expression? And there was a paper that indicated that CDC27 haploinsufficiency was reported to suppress chromosomal instability. So if the targeting was going on without some compensatory action, one would expect that to actually suppress the chromosomal instability. So the next question we wanted to address was, how might the miR-186 overexpression be, what's the mechanism, potential mechanism behind that? So as I mentioned early in the introduction, HSA miR-186 is embedded in ZRAM-B2. It's here in the intron in ZRAM-B2. This is a gene map from Gene Bank showing the exons in the boxes and introns as the lines. And so you're probably saying, okay, so what's ZRAM-B2? ZRAM-B2 is a differential splicing factor that has 2 zinc fingers, which are diagrammed here. And zinc fingers such as this have been shown in other systems to be targets for arsenite displacement of the zinc and disruption of function. So our working hypothesis is that arsenite exposure is disrupting ZRAM-B2 function by displacement of the zinc. There's a homeostatic response to that by increased ZRAM-B2 transcription, which results in an increase in HSA miR-186 as a byproduct, potential byproduct, and that results in mitotic dysregulation and chromosomal instability, contributing to the cancer. And so this question was picked up by Dr. Mayuk Banerjee, who joined the lab a couple of years ago as a senior research associate and is now an assistant professor. And so published this paper this past summer, where he looked at disruption of ZRAM-B2 zinc fingers by arsenite. And so these are mass spectra with synthetic peptides, zinc finger 1, zinc finger 2, and you can see that incubation with arsenite clearly induces a mass shift that corresponds to the addition of arsenite. So this is ESI, MS, and the shift of 12 is a function of the 6 sulfhydryls that can change the number. So he also looked at, in an acute experiment, what happens to ZRAM-B2 expression and function in a dose response experiment from 0 to 5 micromolar. And what you can see here is that even as low as 100 nanomolar, he sees an induction of protein, but a decrease in function. So this is looking at the splicing of TRA2 beta, which is another splice factor. So ZRAM-B2 is a splicing factor that splices a splice factor. And this is actually a common theme in a splicing factor world. And so what we see is an induction of protein, but a loss of function, which is consistent with disruption of the function. So in summary, cells compensating for arsenite disruption of zinc fingers of ZRAM-B2 may lead to HSA-MIR186 overexpression. That overexpression plus arsenite exposure acting together can induce structural abnormalities. The MIR186 overexpression and arsenite exposure both dysregulate BUB1 expression, likely causing sac imbalance. And that's the spindle assembly checkpoint. And the spindle assembly checkpoint imbalance may contribute to chromosomal instability. So in conclusion, arsenic exposure induced HSA-MIR186 likely contributes to the chromosomal instability seen in the cancers, i.e. the aneuploidy, and carcinogenesis by inducing an imbalance in the spindle assembly checkpoints that can disrupt mitotic regulation and contribute to chromosomal instability. So we're currently working on trying to confirm this in primary cells. We have a near diploid cell line that we've just transduced with MIR186 overexpression vectors. Primary cells don't have a long and great enough population doublings to do these long-term experiments. So we're doing some acute experiments in those. So I believe I acknowledged pretty much everybody who contributed data that I presented and acknowledged the financial support. And once again, very grateful for having received a P30 Center grant to initiate the Center for Integrative Environmental Health Sciences. I'd be happy to take any questions.
That was fantastic. Thank you so much. Chris, this was awesome. We do have some questions coming in, and I have questions of my own too, but we'll start with our participants. Dr. Jason Unrein has a question. You mentioned earlier in the talk that you weren't seeing cancer develop in the rodents, but it was developing in humans. And do you have a rationale for why rodents do not develop arsenic-induced cancer?
That has been a problem in the field for over 2 decades. As I mentioned, so the Walkies lab established this model with prenatal exposure, and they now use what's called a whole life exposure. So they expose the dams 2 weeks before conception and then continue exposure after weaning by putting arsenic in the drinking water given to the pups. And in that model, they see internal cancers, but still no skin cancer. You can exacerbate skin cancer with arsenic in the drinking water if you also use an initiator like UV light or polycyclics, DMVA or something like that. Or if you start with an initiated mouse like the TGAC mouse, then you can get cancer from arsenic exposure alone in the mouse, but not, it's just, that's been a stumbling block in the field.
Yeah. So it sounds like a mixtures issue in the animal model. I wonder if that's true also in humans.
It appears that in humans, the arsenic exposure alone can be sufficient. So there's studies that parallel some of the Walkie's prenatal studies due to an unfortunate accident in Chile, where a water supply was switched. This is very famous in Antofagasta. They switched the water supply, and the new water supply had very high levels of arsenic in it. And that went on for about 10 years, and then they realized what was going on. And because there was disease, and what they were seeing was cardiovascular disease in kids as young as infants, like 14 months, myocardial infarctions due to coronary occlusion. These are very famous back in the 70s. So they switched the water supply back, but now there's this cohort of people who were exposed either in utero or in early life, in childhood. And they followed these people now for the past 40, 50 years. And these people show an increase in lung cancer. They show an increase in chronic lung disease, bronchiectasis, and things like that, also cardiovascular disease, all the things you see in people who are chronically exposed throughout life.
Wow. Wow. All right. More questions. One of our toxicologists, Dr. Holly Swanson, has a question on she asks, are the mechanisms specific to sodium arsenite? Why or why not?
No. This is a short answer. Up until the early 20th century, Fowler's solution, as it was known, as concoction developed by Dr. Fowler to treat psoriasis and some leukemias and such, is actually potassium arsenite. And that is well established to have caused skin cancers. And that was recognized as early as the 1890s.
Wow.
So, hi, Holly.
Great. There's also questions on from her, and then also one of our faculty, Dr. Kevin Pearson in pharmacology, asking about other skin types. Have you tried using organotypic skin cultures or foreskin?
The primary cells that we tried using were foreskin cultures that we bought.
Okay.
Okay. We have not tried to establish our own.
Okay. So my question, I'm going to go, I guess, back to the exposure. You talked about drinking water as one of the main sources, and then we see all the lesions on the skin. So is that a contact from water, or what's going on in ingestion versus contact?
So skin is the major target organ, probably because of the high content of keratins, so the sulfhydryls. So arsenite likes to bind to vicinal sulfhydryls, and skin cells have a high content of that. So that's what's believed to be the targeting. But it also affects all the internal organs as well. I mean, if you give a mouse arsenic in their drinking water and then take their organs and look to ICP-MS, you can find arsenic everywhere, including in the brain.
Wow. Wow. Are there other questions coming in, or anyone have other questions for our speaker?
Oh, sorry, my wife is blending food upstairs at a perfect time. But so my question about foreskin is because of you could probably keep it alive as a tissue, right, for 24 hours or something. So, I mean, I've done, unfortunately, a lot of work with foreskin over the last 10 years or so. But I mean, it's a pretty easy tissue to collect, at least at our hospital, because they do the circumcisions pretty much the same time every day. So it's pretty easy to set up, and then you could do some kind of almost like a 24-hour treatment, I bet, and keep that tissue alive. But anyway, I was just kind of thinking, and to do the primary fibroblast isolation is really easy. If you ever wanted to set that up, like in order to do some translational studies or something, if you had a population or something you wanted to study. But
yeah, so the exposures need to be chronic, which is the fundamental problem with primary cells. We tried to see if we could pull some stem cells out, but we weren't successful in getting enough out to do anything with. So that's a possibility, is maybe in neonatal foreskins, one could get enough stem cells out to establish stem cell cultures, which would be more or less permanent. And that could be very interesting.
Chris, those organotypic cultures, you can culture those for about a month.
Yeah, but we're going 6 months to get them
Oh,
transformed.
6 months. Oh, well, that's a different story.
So Anna tried valiantly transducing the primary cells and growing them out, but they all petered out after about 16 weeks.
Have you looked into some of the skin grafting, you know, grafting the human onto the mouse models?
No, I've got my hands full just with the in vitro models.
Okay, all right.
It's an idea.
Yeah. So Chris, any therapeutic options are there, since you're looking, you know, molecularly? I always like, okay, what's the target? What's the drug potential?
Well, so the therapeutic, trying to cure it after the fact, I think, is probably somewhat futile, other than excision for skin cancers. You know, the internal cancers, another problem. But in terms of prevention or mitigating effects, I think zinc supplementation might be the way to go. So it's been done in vitro by 1 or 2 groups where they've supplemented zinc and where they're looking at specific targets and shown that that helps prevent the disruption. We've got an experiment in progress looking at ZRAMB2 to see if we can prevent the disruption of the splicing. And I could give you a whole nother talk on arsenic disruption of splicing. We're putting that paper together right now to send it out. It induces, even at this very low level, induces massive changes in splicing. I mean, you know, over 1,000 different targets. Yeah, it's amazing.
Wow. Well, this has been phenomenal. I don't know if everyone can unmute or unmute, Zoom, show your camera, and help join me in thanking our speaker for coming all the way.
All the way.
New world.
So
To Lexington.
hey, you never know, those electrons might have traveled around the world.
That's right. Thank you. Thank you. Thank you. Thank you.
My pleasure. My pleasure. Good to see some familiar
Fantastic.
faces again.
Yes.